All right, so I'll get started. We have no major announcements this week except for daylight savings time. We're observing the US change in time. So that means for this session, and I believe also next session, it will be a different time in many places in the world. Just 1 hour, what is this, 1 hour before, I believe? Yes, 1 hour before the regular time. And I think that's the only announcement. Do you have any, Magnus? No? Okay. So then I'll introduce our wonderful next speaker, Dr. Megan Ken. So we're continuing on our trajectory from last week about talking about experimental structural biology with RNA and new ways to think about, where we should put in our predictive knowledge and where we should try to improve our predictive skills. And a very exciting work by Megan. So she got her MD PhD over at Duke in the lab of Hashim al-Hashimi. So studying NMR, which is something my memory is correct. I don't think we've actually had a speaker directly talk about NMR. So that's also new for this group. And she studied RNA actually in the cellular environment, the dynamics of RNA in the cell, as well as RNA small molecule binding. So that is something as well that our group is interested in, predicting small molecule binding to RNA. Throughout her PhD, she was involved in a lot of cross-disciplinary collaborations, doing NMR spectroscopy, as well as computational screening, and biochemical and virology was all things that she's been able to be exposed to and learn about. And now she's over at Scripps, so she came to California, and she's just starting up her lab now, investigating viral RNA-protein interactions in cells and how to target these viral RNAs with antiviral drugs. So we are very excited to have Megan here to talk about her work. So go ahead and share your screen, Megan. And for questions, please either raise your hand or write them in the chat, and we will let you know if there's a question.
Awesome. Thank you so much. Yeah, thank you again for inviting me, and it's been really wonderful to be a part of this group over the last few months. And so I'm really excited to get to talk to you all today, tell you about sort of what I did in graduate school, and then I want to spend some time talking a little bit about my plans as well for my independent group, and that's going to involve a lot of structure prediction, and so I'm really happy to get this opportunity to share that with you and get any feedback. So, any and all feedback is very welcome. So as Rachel said, I'm not going to talk as much about the small molecule sort of stuff today, but I do want to tell you about what we found in terms of the role of RNA dynamics in cellular activity. And so I don't need to tell this group that most RNA-mediated cellular processes are the product of biomolecular recognition. So this can be RNAs to metabolites to small molecules, RNA-RNA interactions, or RNA-protein interactions, which is what I'm going to be focusing on today. And so normally when we think about these biomolecular interactions, we think of an equilibrium like this comes to mind, where you have the two molecules interacting, and then in equilibrium with the bound structure, and the free energy of this interaction being represented by the contacts that are being made, like hydrogen bonds, stacking interactions, things like that. But there's actually a whole other side to this equation that we don't think about as much, which is the conformational penalty. And so what this represents is essentially the energy that it takes to form the bound conformation from the free ensemble. And so actually the total free energy of binding, which is reflected in the affinity constants that we measure, is a linear combination of both the sort of positive free energy that comes from making those contacts, as well as the negative free energy from this conformational penalty. So we know a lot about how to get the information about the contacts. We have all of these wonderful structural methods, crystallography, cryo-EM, things like that, where we can see exactly what's happening within the bound structure. But the penalty is something that's a little bit harder to measure and define. And essentially the best way we have of doing that right now is using NMR. So again, if we think about the free ensemble of an RNA or any biomolecule in terms of these conformational landscapes, most of the time the bound conformation is very, very low populated. It can be even less than 0.1%. So how do we detect that? And as I said, we have to really do that using NMR. And we were able to, and sort of as we go through the talk, and I'll be talking about the energy of the penalty. What we're really talking about essentially is the delta G associated with the population of the bound state, again, in that free ensemble. So this is, fairly difficult to do and not very high throughput. So, why does this matter? Why should we spend time, trying to understand these penalties and these low populated conformations? And to illustrate that, I'm going to sort of go through a hypothetical example, which is, say you have your RNA here that has sort of a very high range of dynamic motions, and it binds to this small molecule in this linker region, this green linker region, and it creates these hydrogen bond contacts shown here in blue. Now, in this case, you can see that the bound conformation is a relatively low population of the total free ensemble. So that means that there's going to be a high penalty associated with this interaction and a correspondingly low binding affinity. So now say you make a mutation to this linker region such that it affects the ensemble dynamics, but it does not actually affect the binding of the small molecule. So in this case, our hydrogen bonds are exactly the same, but you can see now that the bound conformation has a much higher population within the free ensemble. So then just by making this mutation, we now have a much lower penalty and a correspondingly high affinity. And so what this means, practically speaking, is that you can have crystal structures of a small molecule bound to to two different a mutant and a wild type of a biomolecule, and these hydrogen bonds can look exactly the same, and yet they might have very, very different affinities. And so it's really hard to tell just from those structures why that is. And to give sort of a clinically relevant example of that is this really wonderful study from Dorothy Kern's group looking at Gleevec specificity. And this is Gleevec or imatinib is a drug used to treat chronic myelogenous leukemia. And the way that it works is it binds to this BCR-ABL tyrosine kinase, and it has a 3,000-fold difference in affinity between its target kinase and the wild type SRC kinase. But when you overlay the structures of these bound, you can see that it's the bound structure is very, very similar. And I sort of blew this up here so you can see exactly how the small molecules in the same conformation. And so they in order to ask why this is, they did several NMR experiments looking at the free ensembles as well as the kinetics of binding, and they found that in fact this is due to the induced fit conformational change that happens when it binds to BCR-ABL versus SRC. So essentially the global conformational change of the rest of the protein, not necessarily the binding site, and the kinetics of the on rate versus off rate is responsible for this massive change. So keeping all of this in mind as to why, a lot of these things are important, we wanted to ask, are these conformational penalties important for RNA function? And is it important not only for binding, but also for its actual function in the cellular context? And so to ask these questions, we used HIV-1, the transactivation element, or TAR RNA, in order to ask these questions. And so this is a secondary structure of TAR over here, as well as its tertiary dynamic ensemble that was again determined via NMR. And this is our picture of the life cycle of HIV. And just to orient you, TAR is important for this transcription phase. So after the viral RNA has been incorporated, transcribed to DNA, integrated into the genome, and now TAR is responsible for transcribing the RNA for further downstream functions. And so if we zoom in on exactly what is happening in this transcription, The function of TAR is essentially to bind a protein complex, which is composed of the viral protein TAT, as well as members of the human super-elongation complex. And what happens is, the human RNA polymerase stalls after transcribing TAR, and then upon binding, the SAC complex is allowed to phosphorylate the polymerase, which then allows it to continue elongating the viral genome, dissociate, recycle, and create many, many genome copies. So that is what that's what TAR does. And we also know from other mutational studies that actually the actual sequence identity of and the structure of TAR is really only important for this first binding step. After it performs the binding interaction, the actual identity of TAR is not important for the rest of this process. And so that's nice for us as a model system, because we can measure what happens downstream and manipulate the RNA and know that we're really isolating just this one step by changing the RNA identity. And so, and then, additionally, the other nice thing, looking closer, is that we already know a lot about this system. So we know so much about the bound state of TAR. There's been a lot of wonderful work done by many groups, including Jamie Williamson, who I'm now collaborating with at Scripps, looking at how this arginine sandwich and arginine fork motifs are really important for binding to TAT. And additionally, we know a lot about the penalties as well. So again, my advisor, Shima Alshimi, has fully experimentally determined the ensemble of TAR. So with this in mind, thinking about what are the actual penalties that are at play. So again, we want to look at these penalties, what is happening here. And so in order for TAR to assume the bound state of the base triple, it needs to undergo a stacking conformational change from the bent state, which is where it lives in primarily, to the stack state. And then once in the stack state, it also needs to undergo a change to this specific base triple conformation, where one of the bulge residues flips up and forms a base triple platform. And I'll show you more in detail in a moment what that looks like. And so this stacking interaction and this triple interaction create the two penalties that are important for this equilibrium. And so in order to probe these two different conformational penalties, what we wanted to do is make a library of mutants that specifically manipulated these penalties without affecting the region of binding to the protein. And so to look at stacking, we made a series of TAR variants where we replaced the UCU bulge with a U bulge that ranged from 0 U residues to 7 U residues. And again, the idea is that the TAT does not directly interact with the bulge. And so this should change the ability of the RNA to stack without changing its ability to bind the protein. And then similarly with the base triple, again, here's the actual sort of secondary structure of what that base triple looks like, where you have your A27U38 base pair, and then you have the U23 flipping up to pair with the Hoogstein face here. And then so we modulated this base triple with two different sets of mutants, one by flipping the A27U38, and then one by creating a diasa A27, so that one of these hydrogen bonds is disrupted here. And so we made all of these base triple TAR variants for all of the stacking variants. So there was about 29 mutants in total. And so now, let's make a prediction about what these mutants are going to do. So we wanted to start by just doing an in vitro binding assay. And the closest thing we have to that is this, what we call the TAT arginine-rich motif peptide, which is the RNA binding region of the TAT peptide. And so, if we write out this equilibrium, the binding energy of the peptide is going to be equal to, the penalty of the stacking, the penalty of the triple, plus the positive energy of the contacts. And so when we make our mutants in the bulge region, we see in theory, right, the stacking penalty should be affected, but the triple formation and the contacts should not be. So this leaves us with a prediction that the change in free energy of binding between the wild type and the mutant should be equal to the change in free energy of the stacking penalty, again, between the mutant and the wild type. So, so this is our prediction prediction, and now we went to go test it. And so to test it, we have to measure the peptide binding and we have to measure the penalty. So let's start by measuring the penalty. And so in order to do this, we again used an NMR method, and this was developed by a previous grad student in the lab, Don Merriman, and she found that for TAR in particular, there are these two residues, A22 and U23, that directly report on stacking. So this chemical, these chemical shifts here in orange, these are their positions in the most possible stacked state, and then they shift over to these blue positions as they become less stacked. And so and then the expression that we use to calculate the free energy from this is essentially just the log of the population of the stacked state, which is what we get from here. And so so we went ahead and wanted to use this method, but since this was published, we had since come up with a better way to determine RNA ensembles in the lab. Another student, Honglu Shi, used FARFAR, which you're all familiar with, along with NMR to create these higher accuracy ensembles. And just to quickly go over that, essentially, you know, again, they took the secondary structure of TAR, created a conformational library using Rosetta FARFAR, and then selected from that library to fit these NMR measurements called residual dipolar couplings. And then from there, they got a dynamic ensemble and then could do chemical shift validation through quantum mechanical methods such as DFT. And so the ensemble they got for TAR from that, as you can see, the agreement between the measured residual dipolar couplings and the predicted RDCs from this ensemble, the FARFAR NMR ensemble, were in very good agreement and had quite a low RMSD of 3.1 hertz. So Honglu did this not only for the wild type ensemble, but also for the U7 TAR ensemble. And so in order to, again, validate this method, we said, okay, let's take these new ensembles and essentially count the number of stacked versus bent conformations. And we did that using DSSR and see if the populations that we get from these ensembles using this method match the populations of the stack state we get using our NMR CSP method. And so, and again, we do that because this whole process is still takes quite a long time, and so for the almost 30 mutants, it's much faster to do the NMR CSP method. And so just looking at the data here, for a while, the solid bars are the FARFAR NMR method, and the open bars are the chemical shift perturbation method. And we see these are the populations of the stack state for wild type and U7. And you see that there is definitely a difference between the two methods, but that difference is somewhat systematic between the two. So again, what we're going to be ultimately plotting is the difference in the free energy between mutant and wild type. And so when we look at that for U7, this is the delta delta G of the stacking penalty between U7 and wild type. And you can see that the difference is less than 0.1 kcal per mole. So we felt confident, again, moving forward with this method. And so using the CSP method on the mutants, here's the actual data. So this is just for the wild type based triple, where the only thing we're modulating is the population of the stack state. And so you can see here we have a nice trend where you know, the lower bulge lengths are more stacked. And then as we increase the bulge length, we go to less stacked. And then if we look at the mutants in which the base triple was disrupted, we can see that that really has no effect on the stacking. And again, we would expect that because that base triple conformation is important for binding, but it doesn't actually affect the stacking of the free ensemble. So this is sort of in line with what we had predicted. So then moving on to the peptide binding, to do this, we used a fret-based assay. And again, we have that TAT arginine rich motif peptide, which is the RNA binding region. And the way it works is that when it's bound, the two fret partners are are pulled apart and create a signal. And then when it's not bound, they quench and there is no signal. And so this is just an example of what the data looks like. We got these very reproducible KDs for all of our mutants. And then again, here's the data shown in aggregate. So from these KDs, we calculated the delta G of the peptide binding between the mutants and the wild type. And then here, interestingly, so if you look at U2 to U7, we have essentially the same pattern that we saw with stacking. U0 and U1 obviously are outside that. They all of a sudden have really, really low binding affinities, even though they stacked really well. And this makes sense because, again, now that we're actually looking at binding, the base triple is really important. And the U0 cannot form the base triple. It doesn't have the U23 base. And U1, even though it does have a U23, it's constrained such that it's not able to actually flip up and make the base triple. So again, this is in line with our expectation. And then when we look at the mutants in which the base triple is disrupted, again, the prediction is that they will not bind very well, because as we can see with U0 and U1, they don't bind very well. And again, we see that is the case here. So, so again, back to our prediction, we thought that these delta Gs for the mutants that can form the base triple, that is, should really be have a linear one-to-one relationship with a slope of 1 and a y-intercept of 0. And when we plot that, we see very good agreement between these data sets and very good agreement to that prediction. And so you see here we have a Pearson correlation of 0.97 with a small RMSD to the model fit as well. So so this was exciting, and we were like, okay, so this works for in vitro peptide binding, cool. But does this actually mean anything in the cellular context? And so we wanted to move on and actually look at the free energy of the cellular process, which is that transactivation process I showed you before. And in order to do that, we used a luciferase reporter-based assay. And so the way that works is we transfected a set of plasmids into HeLa cells. And so the first plasmid is one in which TAR drives the expression of firefly luciferase. And then we have a constitutive promoter driving the expression of TAT, as well as a constitutive promoter driving the expression of RLUC. And this is a transfection control because it should be independent of the TAR activity. And so just to visualize what's actually happening in the cell, we have that same interaction that should be occurring, the elongation and dissociation. However, instead of creating biogenome copies, we're creating mRNAs that translate to firefly luciferase, which then is a signal that we can directly measure. And so I don't have time to go into exactly how we decided to do these calculations, but it's in the paper. We can talk about it more later, but under a set of assumptions, one of which being that this process is at equilibrium, we're able to calculate a delta G cell value from these different luciferase measurements. And so again, our prediction would be, or I'm sorry, so just to then show you the data here again, so this is the delta G cell for all of our mutants. Again, we see this trend with the lower bulge mutants having more transcription, so higher activity than the wild type. And then as you increase the bulge, the transcriptional activity goes way, way down. And then again, we see for U0 and U1 sort of that opposite trend of also having lower transactivation, which again is likely due to the fact that it's not able to form the base triple. And so again, our base triple mutants, we similarly see a poor level of activity. So again, let's look at the agreement then between the in vitro peptide binding and the cellular activity. And we see here again that, not quite as good as before, but we still have a relatively good agreement between these two data sets as well as to the model fit. And so this is the data in aggregate shown for all of our base triple competent mutants and their delta Gs for these 3 different assays. And so we can see that there's relatively good agreement across all of these assays. And again, that's really significant because, again, for example, the stacking, these measurements are made completely in the absence of the protein, and yet they're still quite predictive of what's actually happening in the cellular context. And so this is just sort of an illustration of the model that, came out of this work, where, essentially if we understand the penalties at play and sort of what's happening in the dynamic ensemble, then we can actually predict what manipulations of that ensemble should do, right? So if you're able to stabilize the bound state, you're going to increase your cellular activity, and vice versa. So then moving on from this to thinking about what I wanted to do in my independent group, where does this lead us, or where do we go from here? What's the bigger picture? And so this is sort of a 40,000-foot view, basically science fiction at this point, but bear with me. And so I think, the ultimate goal is like, what if, we could take an RNA, if there was a virus, epidemic, pandemic, and, we knew the viral genome and there's a new variant or new mutation. What if we could take that mutation, or even predict mutations before they arise? We could completely computationally, or perhaps with minimal experimental data, calculate the penalty, calculate the contacts, and then we can do this with small molecules as well and predict the contacts of potential small molecules. And then we could get data immediately from that computational model about, drug susceptibility, about recognition, about transmissibility, et cetera. And then these could inform public health measures and drug targeting efforts, even before, the mutant spreads. And so this is a very high level, sort of goal. How do we get there? The study that I just told you about essentially answered the simple question of, are conformational penalties predictive of cellular activity? And in this very, very controlled, very well-studied model system, we found that, the answer for this case was yes. So how do we expand this and try to move a little bit closer to that ultimate goal? I think the next question we need to answer is, what biophysical properties are most predictive of cellular activity? Most RNAs are much more complex and have many more elements than TAR. And so I think we need to understand how those different elements in these more complex systems contribute to the overall activity. And so in order to do that, I think we need to look at a more complex RNA model system. We need high throughput experimental platforms to be able to get measurements on, thousands and tens of thousands of mutants, as opposed to just, 30. And then structure prediction is obviously going to be a huge component of this, especially if we want to be able to create fully computational predictive models. And so my plan to execute this, again, now that I'm here at Scripps, is, well, actually, I'm sorry, I'm just going to again go through sort of how these, reiterate how these approaches are different from what I did versus what I'm planning to do. So again, in the TAR study, we already had a very deep structural understanding of the ensemble, and from that understanding, we were able to make a prediction. We then did experimental measurements to test that prediction, and then we got a result, right? And so what we want to do now is, it's not really feasible to gain that level of structural understanding using NMR for every RNA. So we need to take a different approach to be able to do this in high throughput. And so again, when we're thinking about which dynamics are important for cellular activity, in this case, we're planning to first do lots of experimental measurements. From all of those experimental measurements, we then want to model these RNA mutants using structure prediction methods. And then by modeling the experimental measurements with this computational prediction, we can then gain a better understanding of the system and then make further, more specific predictions that we can test. And so we're planning to do this using staying in HIV, but using going to a different aspect of the life cycle. So before we were looking at TAR and its role in transcription, and now we're going to look at the entire 5' leader of the untranslated region of the HIV genome and its interaction with GAG. And this GAG leader interaction is responsible for the packaging of the virus. So the assembly of the core structure, and then transport to the lipid membrane for budding and release. And so Structurally, this is a sort of a cartoon of what the 5' untranslated region looks like. So here's TAR, but now we have all of these other elements as well. And these elements have many different functions in the virus. So like, for example, this green DIS loop is the dimerization initiation site. We have the splice donor site. And then this Psi element is what's one of the major elements that's important for packaging, though a lot of these elements do play a role. And so obviously, the secondary structure doesn't look like this in the cellular context. It actually folds to form a more complex secondary structure that is actually a conformational switch. So in this conformation, we have the dimerization initiation site folded in and forming a tertiary contact with this U5 sequence. And in this case, translation is favored because, again, this dimerization site, which is needed for packaging, is sequestered, and you have the AUG relatively open and available for translation. The other possible fate in the folding pathway of the 5' UTR is this conformation, which favors dimerization and packaging, which is, again, what I'm mostly interested in. And so in this case, again, we see that the AUG sequence is sequestered now to that U5 sequence, and then we have the dimerization initiation site available for the genome to dimerize and then be packaged. And so so what we want to do then, of course, is make a lot of mutants of this RNA to probe these different structural elements. And one of the most important elements to look at really is sort of everything after this poly A site. Mutational studies looking at sort of larger aspects, regions of the sequence have been done and shown that these are all the elements that are most important for packaging. And you'll notice that again in this conformation, we have a really interesting secondary structural element, which is this tandem three-way junction, as you can see here, that are made by these 3 helices. So that is something that we're going to be looking at. And then another area of importance to draw your attention to is this stem of the DIS loop. And GAG initially binds, it's thought, though not a ton of work has been done, but all evidence currently points to GAG binding to this region with the highest affinity. And then after it binds here, it actually oligomerizes along the scaffold. So again, another aspect of complexity in this system is that, we have multiple binding events as well. It's not just a single binding event. Okay, so so if we're thinking about what is actually packaged, we have 2 genomes that are going to be connected in this way. And so we want to make a library of now going up to order of 10 to the 5th. So a 10,000 mutant RNA library. And instead of, again, sort of picking these mutations randomly, we want to systematically introduce mutations to probe specific areas of interest. And so there are a lot of different things that we can look at. So first of all, we can take these individual helices that exist and do essentially what we did with TAR. We can change the loop and bulge sizes. We can change the length of the stem loops. We can change the stability. We can then, also now look at altering secondary structural elements, such as these, different three-way junctions. So we can change the geometry. We can change the stability again of these elements and the arrangement of them. And then lastly, sort of the other broad category of things that we want to look at are these tertiary interactions. And again, we can sort of modulate these interactions, the length, the strength, the kinetics of these interactions. So we want to construct this library in order to probe all of these different properties. And then once we have the library, we want to do essentially, we want to create a really high quality data set that can be really useful, again, when doing our modeling and structure prediction, which I'll talk about in a second. And so the 3 sets of experiments that we want to do with this 10,000 mutant library is first biopackaging. So that the most downstream cellular function of the RNA-protein interaction of interest. We also want to develop an in-cell binding assay. So can we actually get quantitative KDs or something close to that in the cellular context, as opposed to just in vitro binding? And then, of course, to do the in vitro binding as well. And so the in vitro binding will likely be doing in collaboration with Dan Hirschlag, who has the RNA map platform, which I'm sure a lot of you are familiar with. And then these assays we're going to be sort of developing in-house. And so just to give an example of what that data might look like, if we take all of the mutants and, plot the packaging for versus the in-cell binding, for example, we're going to get a mess, right? Like there's going to be tons of data everywhere. And so I'm going to talk about how we're going to stratify that to get more meaningful results. But just at first glance, the other nice thing about this is that it is hypothesis generating, because you're going to have outliers. So, for example, if we have these mutants over here that, for example, say, bind really well, but don't package very well, that's hypothesis generating. We can think about why that's happening. Maybe it binds really well, but it prevents oligomerization. And then, over here, you have the same types of things. So not only are we excited about doing these types of experiments from a structure prediction and modeling aspect, but also from a biological and hypothesis generating aspect as well. And so then, of course, if we think about then looking at comparing in-cell binding versus in vitro binding, this very obviously informs differences in RNA structure and function in vitro versus in cells, which is a topic that is obviously very hot and is being studied from a lot of different approaches right now. So we'd be excited to contribute to that conversation. And so then again, what do we do about the actual structural modeling? Because that's the most important part, right? Really is knowing what the ensemble looks like so that we can actually, provide meaning to some of these results. And so previously, when we looked at the penalties versus the binding, we got this information from NMR. And as I've been mentioning, that's really hard to do. It's basically impossible to do at the moment for a library on the order of 10,000 mutants. So how are we going to do that? And this is where the structure prediction comes into play, and where I'm excited to learn from you all about how to best do this. And so the idea would be that we would be able to use tertiary structure prediction to look at all of these elements of the different mutants. So to look at the angle of stacking, to look at the twist angle for the various helices, to calculate the energy and the stability of the various helices, and to get all of these structural correlates that we would like to get from NMR, but we really can't. And so, again, to kind of visualize what this might look like is that if you have these structural correlates from structure prediction, then all of a sudden, you can start to see patterns emerge. So this kind of looks like nothing, but then, perhaps when you're looking at just the mutants that affect the through-A junction, which again, we determine that, from the structure prediction, maybe all of a sudden there is a correlation, and these correlations are not always going to be linear, of course, right? And again, if you think about, we wanted to look at the slice donor site bulge length, right? Like all of a sudden you might see that, for example, the in vitro binding is affected massively, but the in-cell binding isn't. So then the idea is to then create a model with a linear combination of terms, or simply train a machine learning model with all of the structural parameters and the experimental data to try to get at this ultimate cellular packaging. And then the nice thing about this too is that we have this test set of 10,000 mutants, or I'm sorry, training set of 10,000 mutants, and then we would have this test set as well, which we can then hopefully feed into the model with the parameters from structure prediction, and then test how well it does at predicting the packaging. So that is essentially all that I have today, and I just wanted to thank my PhD advisor, again, Hashim, and the folks in the lab who helped out with this project, as well as Dan Hirschlag at Stanford, Brian Cullen at Duke, and Ursula Scholl-Skallman at UCF Gladstone. They were all really instrumental in completing the work, the paper that I talked about. As Rachel mentioned, my lab is now at Scripps Research Institute in La Jolla, and we are small but mighty. So Catherine is a really talented graduate student who is working on developing that high throughput packaging assay, and Sean is going to be working on the structure prediction aspect. And thank you to all my mentors and everyone at TSRI, which has made this venture successful so far. And thank you all for your attention.
All right, thank you so much. What a wonderful talk. Um, if people could just raise their hands when they have questions and then or write them in the chat, and I'd be happy to read them out. Looks like Horatio. Go ahead and
Yes, hello. So thank you for the nice talk, Megan. I have a question regarding your NMR experiments inside the cell. How many nucleotides could you actually measure? Is there a limitation, experimental limitation, or how does it work? I'm doing computational models, so I would like to know a bit more.
Sure. So you're just to make sure I understand. So you're asking how many residues could you measure at once using NMR? Is that right?
Yes, nucleotides in this case, right?
Yep. Yeah, so all the experiments that we did were in vitro. It's definitely possible to do in-cell NMR. That's not, but we haven't really explored that yet. So in vitro, it's an interesting question because you can measure essentially as many as you want, but the nucleotides are going to overlap. So it's going to be hard to distinguish one from another. And so, for example, for the HIV leader, that's about 350 to 400 nucleotides. And you can take a spectra and look at all of those residues. And a lot of them will be able to be resolved. However, to look at a lot of them more closely, you can use different tricks. Like you can specifically label only adenines or only cytosines or only certain parts of the spectra. So you can sort of zoom in and see what those residues are doing in the context of the whole RNA, but without the crowding on the spectra. So there are lots of things you can do. But for RNA to kind of like very easily and clearly see the spectra, without having to do a bunch of extra tricks, I would say like maybe around 100 nucleotides or so.
Okay, great. And just one more question, follow-up question. So in the case of cryo-EM, sometimes, in the literature, they said that the RNA backbone is forming different ensembles, right? Do you see similar things with your experiments?
That the RNA, I'm sorry, you said that the RNA backbone is different in different samples?
Ensembles.
Oh, the RNA backbone is changing in the ensemble, is that right?
Yeah, exactly. This is what they observe.
Yeah, I mean the RNAs are super flexible. So I would say, the backbone is changing a lot. I mean, enough to even change the whole secondary structure of the RNA. Anything that can happen does happen. It's just a matter of how often it happens, right? And I would say for RNA that you do get these more dramatic changes a much larger percent of the time than you would for, say, a protein.
So you pick up the ensemble that it's let's say has a better density and appears more often, more probability in your measurements or something like that.
Yeah, so I mean, this NMR is all done in solution, so it's essentially at equilibrium, throughout the entire measurement. So what you're seeing at these residues is each signal that you get for a residue is an average of the entire ensemble. And then what you can do is you can change the conditions to see how the residues move to understand more about the ensemble. So to get an entire ensemble for an RNA from NMR, you actually have to combine data from lots of different types of experiments. But yes, it's very easy to see all of those changes.
Okay, thank you very much.
Thank you.
great. Chaitanya, I think is next.
Hi, Megan. Thanks for the
Hi.
great talk and all the cool work you've been doing. I, so I took away that conformational propensities obviously determine cellular activity and are related to binding of RNAs. So if we can modulate this, then we can perhaps improve cellular activity, right? But I mean, I was just wondering, right? Like, why do you think natural RNAs have evolved to have multiple states, and do you think that multiple states do play a role in the story here, you know, like it can't just be about locking an RNA into its functional state, right? Like, what are your thoughts about this thing?
Yeah, that is a really excellent question. I have lots of thoughts about that, a lot of which are speculative, but I mean, you're exactly right. Like, there's a reason that we have RNAs that are so prevalent in the cell that do all kinds of things and that they're so flexible. I think, you know, part of that reason is that I think they do have multiple functions, right? So I think probably there are multiple binding partners for different RNAs. Also, RNAs are exquisitely sensitive to salt. And so, you know, I think we talk about in the cell a lot, but like, you know, which cell are we talking about? Because different cells have very, very different cellular environments. So I think as the same RNA expressed in, you know, a cardiomyocyte versus an adipocyte are going to be very, very different and probably have different, then the flexibility allows them to achieve these different conformations, and so allows the cells to do more things with the same piece of genome, essentially, just by changing what's going on in the cell. So I think the ability of the RNA to redistribute. Oh, and then the other thing too, actually, that I would say is also it's a lot more easier evolutionarily to get to new functions if you have an easily reweightable ensemble. So I think perhaps there's some conservation happening there. But your question is a good one too, in that, like, let's look at clinical variants. And so I didn't really mention this, but another thing that I want to do is including in these libraries, you know, what clinical isolates of HIV do we have? And then what is that telling us about, you know, what the RNA is doing in an infection setting?
So something as simple as like salt concentration does change like the conformational propensities as well. Is this very drastic or like subtle changes?
So yeah, so actually they're pretty, they can be drastic. You know, how physiologically relevant that is, you know, a little bit unclear, but I mean, going from I mean, so actually that 7U TAR, I can't remember, I'm struggling to remember which concentrations Don used, but essentially the stack state went from being like 10% in the ensemble in the presence of 0 millimolar magnesium, and then up to, I think, 3 millimolar magnesium, which is a cellular concentration. It went to being 89% stacked. So it's basically turning the ensemble completely around.
Yeah. Okay. That's so interesting.
Yeah.
Thanks for all the insights.
Yeah, thank you very much for your questions.
All right, I have a question kind of going off of talking about salt. So I think that's something that the prediction community talks about a lot and hasn't done much with, right? Is how much should we be paying attention to kind of experimental conditions when we're doing our predictions, slash should we care about where waters are, where ions are, et cetera, in our predictions? So I'm wondering if you could comment on that and kind of weigh, I guess, your two energetic terms of that penalty. So you mentioned it really affected the ensemble, but it can also affect binding, right? And desolvation, for example, could be a large energetic effect. So I'm wondering if you could comment a little bit more about, like, what do you think we should pay attention to as predictors?
Yeah, absolutely. So I mean, yeah, it's hard, and you know, again, I haven't done a lot of structural modeling myself, but from docking molecules to RNAs, you know, it's definitely the ionic components are really, really difficult to model, but they are really important because essentially, you know, for RNA, it's made up of all this massive phosphate backbone that's super negatively charged, and these divalent ions are really dramatically, because they're able to coordinate those phosphates, it can really dramatically change the structure of the backbone and what it's able to achieve. And so I think, you know, I think specifically divalent cations are really important for backbone structure and modeling. And I guess for binding itself, I'm not sure that there are many metals that are coordinated in binding events necessarily, but just the fact that it changes the geometry, that what's available, what residues are available for the protein to bind, I think that would be the case. And then, you know, another thing that came up from Honglu's work looking at like MD simulations and things, is that often the sugar pucker of RNA can be modeled incorrectly a lot of the time in a lot of these simulations, and that also has to do sometimes with the ionic coordination. So I think sugar pucker is also something to maybe pay attention to more than we might think about.
Thank you.
I wanted to ask about the like the prediction methods. So you mentioned, I think it was FARFAR. So have you tried other methods, and have you tried molecular dynamics? Because I think to some extent you don't really have to start from sequence to get to 3D models. You more or less know what kind of molecules you investigate, and you kind of want to understand more the dynamics that to some extent molecular dynamics would be the perfect tool to use. You mentioned already some problems with molecular dynamics and sugar puckering. So yeah, I wonder what you think about using molecular dynamics for that. And of course, this is super expensive. So of course, for thousands of mutants, yeah, you cannot really use that, but maybe there are some ways to do it.
Yeah, I think you're exactly right. I think in terms of molecular dynamics, the one of the applications I think that's really helpful for is looking at tertiary interaction strength. So I think and sort of what happens with some of those key interactions. And so I think maybe even modeling part of the RNA, perhaps, to kind of reduce the computational expense, if there are specific questions that we're interested in. And again, I think tertiary context is one of those things we would want to look at. But like you said, we can't run full simulations for all 10,000 mutants. And then in terms of structure prediction, yeah, I mean, I want to try all of the programs, right? So, I mean, I kind of, there are so many folks making really great programs in this group. And so I think one of the first things I want to do when we start working on that is to sort of benchmark all of these amazing programs that are out there for our system and see, sort of what works best, and perhaps even, model, make multiple models with results from the different, programs. And that's the thing too, is I also, if for people who are mostly dry lab and wanting experimental data sets, like that's kind of also how I see my utility to this community, is making those data sets that can then be useful for people who are developing these algorithms.
Is there more questions from the audience? Feel free to type questions as well if you are not able to use your microphone right now. I have one last question, and then we'll see if any other questions come in. So for the 5' leader of HIV, how do you see tertiary structure prediction playing a role? And what I mean by that is, do you see mostly it predicting individual stems or individual, pairs of stems, or is the aim actually to have prediction be accurate on the entire structure?
I think it's really to be accurate on the entire structure. I think the individual stems are important, and there are definitely things I want to look at there, but I think it's the geometry of the larger pieces of the structure in relation to each other and what those distances are. And I think especially for thinking about GAG oligomerization, like the question is really, what larger scaffold is this RNA making, and how does that play a role in how GAG is able to bind and oligomerize? So I definitely think that is more a tertiary level question. However, I do think the individual helices are important too. And well, I didn't really mention drug targeting at all, but sort of the other piece of what my lab is going to do is targeting RNA. And so we're going to target the leader as well, so that hopefully can be a lot of crosstalk between the results that we get on each side. And part of the ways that we want to do that targeting is by stabilizing non-native conformations that GAG doesn't recognize. And I think in that case, both understanding the global structure and how, in the sort of the kinetic landscape of how it exchanges between those larger forms are important. But also, I think that's also somewhere where the structure of the stems themselves could be really helpful, because by even eliminating a bulge or changing a loop size, that can be dramatic in how the protein is able to recognize the RNA or not. Looks like
All right,
there's.
Yeah, if you want to, I can read it out so everyone can hear. So the question is, among three sites of RNA, so the three different parts, the sugar, the phosphate, and the base sites, wondering if you can comment on the most important for determining tertiary RNA structure, including dynamics in your case, based on your experience.
That's a great question. That's also something I'd be curious for your all of y'all's thoughts on. I'm not sure. I want to, I think for tertiary structure, the phosphate backbone is really the most important thing, and especially if we're thinking about how larger pieces of the structure are oriented relative to one another, their interaction, again, like I was saying, with those cations and how the phosphate backbone is able to fold is really crucial. I think thinking more about local structure and like binding sites and active sites and things like that, I think the nucleotide orientation in that case is super important. I mean, even just a tiny shift in the planarity of a base or how much is flipped in or out can also affect binding quite a lot. So I guess that's what I would say, but I would also be curious to hear what others have to say.
Any other questions or comments from anyone in the audience? All right, there is none. I'm going to say thank you so much for giving a wonderful talk. I think you've exposed our community to the type of problems that we can help solve, right? I think a super big challenge, 5' leader, is like a huge prediction challenge, right? But certainly an interesting one. So thank you for coming. And as always, the recording will be published on YouTube in a couple of days when I get to it. All right, thanks everyone.
Awesome. Thank you so much.